Hey, this is Calvin. This is a video about the coronavirus. My approach to stop the spread of the coronavirus, it is a very minute addition to the world. Actually, so much research is going on. This is just one of my small ideas to combat this virus. So it's a very minute contribution. So as I'm Calvin Montfort, and I'm targeting angiotensin converting enzyme 2 of the lung. Okay, my goal is to point out the mechanism of angiotensin converting enzyme, to start with the physiological role of this enzyme, to explain the route of entry of coronavirus, my hypothesis on inhibition of route of entry, and this is nothing formal. There are no, I have taken not a single formal standard evidence or uh, standard reference, and this is not a standard reference either, as I always say. This is a hypothetical channel, and hypothetical channels deal with hypothesis. So this is a hypothesis. So let's look at the structure of angiotensin converting into enzyme 2. Angiotensin converting enzyme 2 converts angiotensin 1 to angiotensin 2. Angiotensin 2 produces aldosterone from the adrenals, which retains water. So we can see that there is an S1 and there is an S2. So I'm sorry, there are two S1s. There are two S1s, and these two domains are where all the drugs and angiotensin 1 drugs and even the virus can bind. So this binding site is the substrate of the enzyme, substrate 1 and substrate 1 again. These two substrates can be exploited by the virus to enter. Um, I don't find any reference showing that the substrate is where the virus enters. The virus may enter at any point in this enzyme. It may enter from tyrosine, glutamine, proline, anywhere. So histamine, anywhere, any part of the enzyme. So by using steric hindrance and partially binding this enzyme, we can inhibit the entry of coronavirus receptors by steric hindrance. I'll explain a little more. So I'll explain the mechanism. The reason why such an enzyme is present. The oral mucosa, kidney, and lungs have angiotensin converting enzyme to detect thirst and convert angiotensin 1 to angiotensin 2, which retains water. Lungs are very sensitive to water level to prevent edematous lung, the swelling of lung. The virus uses the enzyme angiotensin converting enzyme 2 as receptor for the entry into the type 2 lung cells. Okay, let's imagine a man blowing three balloons together with this effort. The balloon coated with soap water is easier to inflate. Similarly, work of breathing is easier in alveoli of the lung that has surfactants like soapy material. The surfactants producing cells are type 2 pneumocytes, which contain the angiotensin converting enzyme 2, uh, which are invaded by the virus by angiotensin converting enzyme 2 and are destroyed, thus increasing the work of breathing by decreasing the surfactant, thus causing breathing distress. Okay, let's imagine the entire rectangular cell is a type 2 pneumocyte. At the surface of the cell, we have angiotensin converting enzyme 2, and the substrate domains have been marked with the red circles, and coronavirus with the green circle and the spikes. So the coronavirus binds to angiotensin converting enzyme 2 and enters into the cell where it dominates the nuclear host machinery and the DNA, and produces copies of itself and comes out of the cell. Now let's explain captopril, lisinopril, and enalapril. The binding method of these drugs are through substrate of the enzyme. So captopril, lisinopril, and enalapril bind to S1 domain of the angiotensin converting enzyme 2 and block the enzyme, thus preventing aldosterone 1 from binding to angiotensin converting enzyme 2 and converting it into aldosterone 2. Sorry, not aldosterone. Angiotensin 1 to angiotensin 2, thus preventing the binding of angiotensin 1. I'll explain again. Captopril, lisinopril and enalapril, they bind to angiotensin converting enzyme 2 substrate domain to inhibit binding of angiotensin 1 and conversion it into angiotensin 2. So thus the aldosterone production is decreased from angiotensin 2 because angiotensin 2 is not produced from angiotensin 1. Okay, now this receptor can be partially bound with a partial antagonist so that the virus cannot bind due to steric hindrance. These are the substrate domains again. Okay, now I explain my possibilities. We can study the binding method of angiotensin converting enzyme inhibitors. These drugs can be divided into two and selectively bind to the enzyme partly and making a van der Waals sphere of influence to inhibit the binding of viral domain. I mean, this difference between the binding of virus and angiotensin 1 can be studied and made use of. Okay, we can make a ball of missile. I can make a bundle of missiles with missile technology and can be used to encapsulate the drug with lipids along with angiotensin 1 to selectively bind to the receptor and another domain in the same missile bound to a particular antagonist. Angiotensin 1 from the same patient can be titrated. I mean, angiotensin 1 from the same patient can be taken out and when the missile can be made and the multiple missiles coated with angiotensin 1 will directly go and bind to the angiotensin converting enzyme in the lung. So now after it has binded, another domain in the same missile can be used as a partial antagonist and some of the molecules in the angiotensin converting enzyme can be antagonized so in such a way that angiotensin 1 can be bound but the viral domain cannot be bound. This is just my imagination, it may not be possible. Thanks for listening.